So I just wanted to start off with the problem that we have in data science, where data is growing at such a rate that it's too big to practically download and store. And this is a burden on researchers. So the data biosphere is the concept around the data biosphere is inverting the model of how genomic data is shared. The traditional approach is that the data would be brought to the researcher. So the data might be in several locations, for example, one place being the short read archive, and a researcher has to copy and move that data either to their own personal computer or to their institutional server, and that can be costly. For situations where researchers are working with managed access data, that can be a really difficult burden for researchers because it's harder to enforce security. You have to work with your own institution to make sure that you're following the correct protocol for working with that type of data. Additionally, it's just redundant infrastructure, and it's also siloed compute, so it's really difficult for researchers to collaborate either between labs at the same institution sometimes, or especially across different institutions. So what we're trying to do in the data biosphere is bring the researcher to a centralized place that hosts the data. This helps reduce redundancy and costs. It also means that some of the platforms that you're using can help take on some of the security measures that are required for working with managed access data and can provide active threat detection and auditing. There's greater accessibility, so not every researcher might have an institution with a strong cluster or other resources, and so it really limits people's ability to do large-scale analysis. And it's more elastic and shared compute, so it's used more efficiently. So how should this be structured? The ideas that we're bringing to the data biosphere are that it should be modular and comprised of different components. So this means that, for example, Dockstore and Terra can provide different parts of what you might need for your analyses. It's community focused, so many groups that contribute to these projects can foster diversity of ideas and progress. It's open source, so that developers can work together to improve the software and features. And it's standards based, so we're using standards that are being developed by the G84GH, and this allows for new platforms to be easily incorporated into the ecosystem to help this grow. So what is NHGRI as the AnVIL? It is using these concepts that I just previously brought up. It's a scalable and interoperable computing resource for the genomic scientific community. It uses a cloud-based infrastructure. It provides data access and security, particularly for managed access data from the NHGRI, the National Human Genomics Research Institute. Much of the data hosted by NHGRI that is controlled access is on the database of genotypes and phenotypes, which is shorthand dbGaP. And so what we're trying to do here is use the same allow list that dbGaP has for researchers that are granted access to use managed access data, and that we can provide that data through the cloud. It's also a collaborative computing environment for data sets and analysis. So it makes it much easier for you and your collaborators across the world to work in the same workspace and reproduce analyses and results. And we have a strong focus on training and outreach to help you get started, which is part of our workshop today. So just to visualize this a little bit more, in the AnVIL platform, what we're trying to provide is that you have your data. It can either be in the cloud, and it might be hosted in something like Terra, where a lot of current AnVIL data is, or you might bring your own data. And then you'll need tools to analyze that data, which can come from platforms like Docstore or the Broad Methods repository. And those can be easily handed off and work with Terra workspaces, which is the environment where you can actually conduct your research, and that can help make scientific discoveries faster and also increase the scope of the science that you're doing. And this is all currently in the Google Cloud platform. So as AnVIL is not the only platform that's moving to the data biosphere idea, the NIH has several other initiatives that are encompassing this idea. There's the NHLBI Biodata Catalyst, Gabriella Miller Kids First, NCI Cancer Research Data Commons, and not a NIH initiative, but also the Human Cell Atlas is working towards the data biosphere approach. So not only can you analyze data within the AnVIL, but you can potentially compare data from all of these different resources, really scaling up the types of analyses and the samples that you have accessible to do your research. So ultimately, all of these different initiatives are using many different platforms, but one of those is that the Terra workspace. So you can hand off data from all of these different locations into the same Terra workspace to do different types of analyses. The Terra workspace provides not only data, but interactive analyses using Jupyter notebooks that use both Python and R, which you'll learn about today, and batch analyses. And this is where Docstore provides reproducible Docker-based workflows that can be easily handed off into Terra to conduct batch analyses. And like I mentioned before, data for some of these initiatives is coming from dbGaP. So the data will then be hosted in platforms like either Terra or Gen3 that can be handed off into Terra. So with that said, how do you start using the AnVIL? I think the best place to start is the portal, which is the anvilproject.org. There's a representation here. You can learn more about what the AnVIL has to offer, and you can either launch the AnVIL directly in Terra here, or learn a little bit more about the data, tools, and training available. So there's many different user paths into the AnVIL, and we'll demonstrate those more practically today, either starting in Docstore or starting directly in Terra. So with that said, I'm going to hand it off to Louise. We're going to switch screen sharing.
So hi everyone, my name is Louise Kabonsai. I'm a software engineer at the UC Santa Cruz Genomics Institute, where I work on Docstore. So today I'm going to give you a brief introduction on what Docstore is. So what is Docstore? First, I'll give you the full description. Docstore is a free and open source platform for sharing scientific tools and workflows. It's a registry of Docker-based resources described using popular workflow languages like CWL, WDL, Nextflow, and now Galaxy. So our goals are to increase the portability, the interoperability, and reproducibility of research in the sciences. What we mean by portability is that workflows should run in any environment as long as it supports Docker. And for interoperability, like Beth mentioned, we follow the GA4GH API standards and support multiple integrations so that people can actually use their preferred compute and data storage environments. But most importantly, as scientists, we care about reproducibility. So Docstore is a place for researchers and developers to share their work so that others can also use it. The combination of using containers and workflow languages minimize the redundant and error-prone installation of software, but also increases the transparency of the methods used in the actual analysis. So this allows others to verify results and apply existing methods into their own research. But in simple terms, what is Docstore? It's a platform for sharing research software. You can think about it as an app store for bioinformatics. So how is Docstore useful and what problems does it solve? Well, as our first example, let's go over the simplest use case, which is search and findability. So this is a case where a researcher wants to do some type of genomics work, whether that be assembly, alignment, variant calling, an entire genome-wide association study, or whatever they are interested in. But they want to know what software can they use for the job, what works with their data, where do they download that software, and what version of that software should they use. So this brings us to Docstore's function as a centralized registry. So on Docstore, you can search a centralized catalog of bioinformatic tools and workflows. This means that content of Docstore can be hosted from multiple locations. So existing source control repositories like GitHub, Bitbucket, Quay, Docker Hub, etc. And you can even host your descriptors directly on Dockstore. And all these resources are aggregated together in one place on Dockstore to form a centralized library for better user experience and functionality. Let's actually go over to Dockstore and see what this looks like. So when you first get to Dockstore, this is what it looks like. You can sign up with either your GitHub or Google account. I'm going to go ahead and just log in here with my GitHub credentials. And this is your dashboard. When you create an account, you can see the workflows or organizations that you manage. But I want to show you all the different integrations that we have. So like I said, you can hook up your GitHub account to connect your descriptors, your Google account, and all other a bunch of other source control repositories to help manage your data. You can also link your Zenodo account and your ORCID ID to manage and connect to your publications. Right. So in addition to that, Docstore also has a search functionality. So you can actually find descriptors. So once you've connected to the ecosystem, if you are an author, you can bring in your workflows where they might be stored. But if you're looking for some workflows to use, you can actually use a search functionality to just see what kind of things are out there. So you can filter by entry type, whether it's a tool or workflow, by language type. You can try and find some authors, etc. I'm going to go ahead and show you, like, for example, if I want to look for some top med workflows, or if I want to go ahead and look for some GATK workflows. As you can see, there's quite a bit of entries that pop up, over 107 here alone. But so sometimes this can be a little difficult to navigate and find certain things that you're looking for because there are so many results that pop up. So I want to take it back to the Docstore feature, organizations and collections. So organizations on Docstore is a place for groups, labs, consortiums, any type of collaboration, to create a place where they can showcase their projects, showcase the workflows that they've developed for certain aspects of research. These organizations, you can add members with permission-based roles based on who can add workflows or edit things on the collections. And on the collections itself, these are basically playlists or groups of tools or workflows that are highlighted by an organization to kind of organize things they want to present to users. The collections in an organization themselves have their own markdown descriptions. And we can just go ahead and see what this looks like. So this is the AnVIL organization. You can see that these are the workflows that have been added to collections by AnVIL. And you can go ahead and see that the GATK workflows are also here. But if you'd like to see all the other types of organizations that have been created on Docstore, there's quite a few. You can go directly to the Broad Institute organization, for example, and also see the GATK workflows there. Another common reason to use Docstore, not just for searching and sharing, but one of the main use cases that's very common in research is that a researcher might already know what software they want to use, but they actually just have a hard time actually installing that software or getting it to run the way they want it to. So this could arise from a variety of problems. For example, a problem installing the software because it was built on a different system, so it might not compile or build on their operating system or machine, or the installation documents might be incomplete or out of date. Another reason to use workflows on Docstore and tools on Docstore is to resolve some issues that might arise from dependency problems. So software you might be interested in might require a different version of a dependency that's available on your environment. This is really common with things like Java or Python, or you could be using multiple tools that have conflicting version requirements of a dependency. Like maybe one of your tools uses something that needs a certain version of a dependency, but another one of your tools needs maybe an earlier or later version of that dependency. So how do you manage those types of conflicts? Docstore solves these problems by the way it packages its content. So software on Docstore is packaged up in a container. It's packaged up using containerization technology and described using descriptor languages. So a container pretty much packages up the code, the dependencies, and the operating system, which are required to run that software. And this resolves the common installation and dependency problems that I talked about earlier. And the script that actually outlines how to use that software step by step in analysis is written using a descriptor language. And Docstore currently supports CWL, WDL, and Nextflow as a descriptor language. But like I said before, Galaxy is actually now released as a preview of our next descriptor language, and we're currently developing that, and you'll be able to use it soon. So because Docstore packages up software into descriptors and containers, it allows analysis to be moved from environment to environment. If you want to move from clouds to different virtual machines, to different servers, to different compute resources that might be available in your institution, or if those compute resources vary significantly between your collaborators. It should, by packaging up your analysis in this way, you should be able to use analysis in a portable way. So how does this actually look like? So content on Docstore can either be registered as a tool or a workflow. A tool is basically a single container and it performs a single action that's outlined by a descriptor. A workflow, on the other hand, is usually when you're chaining multiple containers together to execute multiple actions or steps, but all of this is still outlined by a descriptor. And tools and workflows are the types of content that you can find on Docstore. To get a more concrete example of what this might look like, for example, you can see BWA-MEM would be an example of a tool. Everything that you need to run the BWA-MEM software or build it is going to be already packaged up and ready to go in a container. And the steps you need to run BWA-MEM will be written out in a descriptor language. But in contrast, an entire variant calling pipeline is actually what is more common of what a workflow looks like. So a variant calling pipeline is composed of multiple steps, which each might use its own different runtime environment that will be each packaged up into its own containers. So things like a step for processing, a step for aligning, and a step for variant calling. One way to look at it is a tool is one, and workflow is many. And just to note here that a single tool, you can register a single tool as a workflow on Docstore, but a multi-step workflow can't be registered as a tool. So this is done for kind of like a modularity purpose. Maybe you want just a specific thing done. But technically, this BWA-MEM example can be registered as either a tool or a workflow. Okay, so let's go down a little bit into what containers, descriptors, and parameter files actually look like. So I've said this a bit before, but a container is a packaged up code with all of its dependencies, so the software can be portable and run quickly and reliably as you move it from one computing environment to the next. And the descriptor is the workflow language used to describe how to run your pipeline. I've underlined the script and descriptor here because that is basically what a descriptor is conceptually. It's saying, it would say which containers you're going to run, what steps to run and when. You're going to also define some parameters for what you need to run your task, which include things like what types of input data should go into your workflow and what should be the output data coming from your workflow. You can include the computation requirements also in your descriptor, so that those computation requirements will either allocate resources when running your machine or even be used to spawn an actual instance if you're, say, launching your workflow into one of the cloud platforms that's integrated with Docstore. And you can also add metadata to your descriptor so that users can know more about how to actually use it and to make your descriptor more precisely reproducible. So the parameter file actually completes this picture. Your descriptor is more of a template that has some variable parameters, kind of sort of fill in the blanks. And your parameter file is actually where you would fill in what specific inputs and compute requirements you need for your particular task. So for example, you would say, I need a BAM file in your descriptor to run my workflow. But in your parameter file, you would say, use this particular BAM to run the workflow. So I'm going to go over a quick, simple Docker file of how a container actually looks like. So first, a little bit of jargon clarification. You might hear the words images and containers used interchangeably. And that's pretty common. One thing to know is like an image is more like how the packaged up code looks like when it's not running. And a container is simply just a running image. So this is how it looks like at rest. And this is how and the container that's actually running and interacting with processes is what the term container means. So images of containers are built from Docker files. Don't worry too much about the specifics of this Docker file. I'm just really trying to emphasize the point that your Docker file describes your packaged up environment, including so what it might be building off of, like which operating system or base, any metadata that you might want to include, like authorship or whatnot. And specifically, it's going to outline what dependencies your software needs to run and be built. And then you're also going to include the actual analysis software in your Docker file. And just a brief example of what a descriptor might look like. So this is a simple little descriptor, the workflow description language. You don't have to worry too much about exactly what the syntax or code means. Just remember the high level point to understand is the descriptor encloses everything someone needs to run a particular task or to complete a particular piece of analysis. And that means it'll have things like what input parameters are needed to run this program, what is the commands that are going to be executed, what is the runtime environment that is going to be used, for things like which containers need to be specified to run this task, and what compute requirements need to be allocated to run this task. You can also include things like metadata, like I said before, which includes authorship, contact information. But you can even include things like what exactly do these parameters mean and what you should do with them. But once you have your descriptor and Docker containers ready, you would register these as tools and workflows on Docstore so that you would have a centralized Bioinformatics catalog for resources. So your descriptors are going to be stored in your source control repositories. You can even store descriptors directly on GitHub, on Docstore, but your images will be on Docker registries. But the biggest point here is they're all aggregated onto Docstore to create a centralized catalog for bioinformatics tools and workflows. And then you can export these workflows into the analysis environments that will contain your data and compute resources to actually conduct your research analysis. And this whole interoperability is made possible through the GA4GH APIs. So what this actually looks like on Docstore is that you can launch your work. So this is a landing page of how a workflow on Docstore might look like. This is specifically the Files tab. And then on the right here, you can select the Launch With environment. We'll go into this later in the hands-on portion, but basically you can launch this workflow into the analysis environment and partner platforms that Docstore integrates with. So this example is a structural variant calling pipeline using graph genomes contributed by Jean Monlong and Charles Markello from the VG team at UC Santa Cruz Genomics Institute. And this is how it looks like once you select the launch into the AnVIL ecosystem. And we'll go more into how to actually do this later today. The workflow we're launching here is the Cumulus workflow, which is cloud-based, single-cell, single nucleus genomics analysis. It's in the AnVIL. It's one of the workflows in the AnVIL organization Cumulus collection, contributed by Bo Li and Yiming Yang at the Cumulus team at the Broad Institute. So the earlier question, what kinds of problems does this solve? Well, this really enables us to have large-scale collaborations, scale to the size of data sets that now exist in genomics, and to be able to reproduce the research that people conduct and share in their publications. So for example, a team of collaborators from different institutions are working together on a study that analyzes the genomes of thousands of individuals. Each institution uses a different compute environment for analysis that could be composed of workstations, local machines, different types of servers, clusters, or entire cloud platforms. Docstore's way of packaging this content makes sure that the pipelines work across all these different types of compute environments that might be used by these collaborators. And the algorithm portability then lets you move and scale your analysis in the cloud as these data sets grow. This enables research to go beyond the compute resources that are traditionally available in your local environments. And the organizations, collections, and the digital object identifiers that you can create for your workflow through integration with Zenodo allow for others to find, share, and precisely reproduce the research done by the collaborators from their resulting publication. So Docstore has many features to help and many tutorials to help get you started in creating your tools and workflows. Or if you'd like to just launch some tools and workflows that are available on Docstore, you can do things like creating snapshots if you do host some workflows, and then create and share DOIs to link to your workflows on Docstore into your publications, create organizations, create checker workflows, which is a more advanced thing about making sure that your workflows can work on many environments. But all this how-to and tutorials are available on the Docstore documentation site. So, and also when you need to get some help on Docstore, you can navigate to our little help icon that's available on the help toolbar on Docstore. that will take you to the Discuss forum that Docstore uses to engage with users and to help people out when they're having trouble. You can talk about bioinformatics, talk about your workflows, and get help with your development. So I just wanted to give a thank you to the many contributors, users, and partners that have come together to create the centralized catalog of tools and workflows on Docstore. We currently have a library of over 700 published tools and workflows. And the diagram on the right just highlights just a few of the contributors who have really built Docstore and published their work onto the space so that it can be available for usage by others, and the different platforms that are available to launch those workflows into. I just want to close it out saying thanks to the Docstore team for all their hard work in making this an open resource, and thanks to our funders for allowing us to do this kind of work. Okay. And I'll let Barry go ahead and tell us more about Terra.
Great. Okay. Hi, everyone. My name is Barry, and I work at the Broad Institute in a group called the Data Science Platform Group. And this group is building a cloud platform for biomedical researchers to access data, run analysis tools, and collaborate with one another. This platform is called Terra. And in the next few slides, I'll go into detail about this platform. First, let's start out with what the learning objectives for these slides are. In these slides, I'd like you to understand the three big components of Terra, and that's the data, bulk analysis, and interactive analysis. You'll also learn about how Terra uses containers for portability and reproducibility, understanding Terra's security compliances, and finally, understanding the costs associated with Terra. Okay, so Terra was designed for the data biosphere as a platform for research and analysis, bridging the gap between the data in the cloud and end users like researchers and clinicians and patients. With Terra, you have a central access for data, meaning you have access to several cloud-based data repositories, and you can also upload your own data to Terra if you like. And here are some examples of the data repositories you have access to. Those include TCGA from NCI, some TopMed and GTEx from NIH Data Commons, along with the AnVIL and NHGRI. You also have access to integrated tools like Docstore, which was mentioned in the previous slides, and the Broad Method repository. They both have workflows. You can import you can import workflows from those two repositories into Terra. You also have access to Jupyter notebooks for more interactive analysis, where you can execute commands and change them in real time. Okay, let's take a closer look at Terra. So what is Terra, and how would users interact with it? Terra is an end-to-end cloud-native platform. And in other words, it's a web application you open into a Chrome browser that enables you to do all aspects of a research project, all in one place. In this slide, we break down a research project into 4 components: find, access, analyze, and share. Terra has features to allow you to do all this in one place. You find and access data. This includes the public and restricted access cloud-based data sets mentioned in the previous slide. You can access data securely and immediately using Google's the Google groups and authorization domains to control access. You can analyze the data using workflows from Docstore and the Broad Method repository and perform interactive analysis through Jupyter notebooks. And then finally, you can share your work with your collaborators or work with your collaborators either individually, with groups, or with everyone on Terra. Okay, so let's explore how Terra does all these things in detail. It all starts with the Terra workspace. Terra workspaces are the building blocks of Terra. They're computational sandboxes where you can access, organize data, and execute analysis workflows, and also collaborate with others. All workspaces have 5 major components to them in the form of tabs. You can see this on the slide. There's the dashboard tab. This is the landing page for a workspace. It gives an overview of the project components of the workspace. You can edit the dashboard in Markdown to ensure your collaborators and others who are just viewing the workspace understand what you're doing in the workspace, understand the workflows that are involved in the workspace, and other details. There's the data tab, which keeps track of all your project data, no matter what size it is or where it's stored on the Google storage buckets. And there's 2 modes of analysis, like I mentioned earlier. There's the notebook and workflows. With notebooks, you can run commands on either a Python or R kernel using Jupyter Notebooks. And with workflows, you can conduct bulk analysis using WDL workflows. Then lastly, we have job history, which allows you to monitor and troubleshoot bulk analysis submissions. So every workflow that you've executed is listed in the job history. All right, so let's take a closer look at the data. So to understand how data is organized in Terra, it helps to view the workspace as a computational sandbox, where you can access, organize, share, and analyze data. And at the heart of the workspace is the data tab, which holds your workspace data tables. These tables can be thought of as spreadsheets that help organize sample metadata. For example, the type of tissue a sample is from, whether the sample is tumor or normal, and more importantly, the links and paths to the location of the raw data, like BAM files or FASTQ files and images. These raw files are stored in different Google buckets. The one within the workspace in this diagram is the workspace bucket, and that's a Google bucket that's always created with the associated workspace that you've created. Sorry. There's also the different cloud-based repositories. So that data library on the right, that is where, for example, TCGA, AnVIL, etc. Data repositories are, and you can access those buckets as well, but they live outside of your workspace. There's also other cloud-based storage not associated with your workspace or with a repository. And these are normally independent Google storage buckets. So Google buckets that were created independently through like Google GCP, Google Cloud Computing Platform. All right, so next we'll explore the different cloud-based repositories. Terra hosts a number of public and restricted access datasets in its data library. Here's a screenshot of a few which you can browse, and more importantly, you can import these directly into your workspace for analysis. The 1000 genome is an example of open access datasets, and those are the top 2 in this column and row. And then we have AMP Parkinson's disease, which is an example of restricted access datasets, which are datasets where you need prior authorization in order to access them, as opposed to the 1000 genome, which are open, and you can directly go to the browser, data browser, and bring in data into your workspace. The main difference is when you're working with restricted access dataset, you'll need to make sure you have authorization with the specific repository. So on their end, go through the hoops of getting access, and then finally linking your Terra account with that repository, so you can easily import datasets into your workspace. Okay, so suppose you're at a point where you've chosen your datasets of interest and you've imported this dataset into your workspace. What do you do next? What can you do with the data? There's two things you can do, and that's bulk analysis and interactive analysis. The bulk analysis, this is how you standardized or best practice pipelines are typically done. If you know the data processing doesn't require much tinkering, like the workflow doesn't need much or changes in the arguments, then you simply can plug in your data into a workflow and have Terra do the heavy lifting of managing executed workflows on several samples. And then there's interactive analysis. This is useful when you're visualizing or analyzing data of any size in real time. It uses the Python and R-based Jupyter notebooks. Typically, the interactive analysis occurs after getting outputs from your bulk analysis workflows. At that point, users would like to use R's powerful statistical and visualizing applications to dig into and interpret the workflow results. As an example here, performing GWAS or doing QC on your samples. So now let's talk a bit more about bulk analysis. Terra helps you streamline your bulk analysis by implementing open and shareable modular principles. This means you often do not have to write your own WDL workflows from scratch. And instead, you can search for published workflows from Docstore or the Broad Method repository and add it to your workspace. Examples of this sort of analysis tools are aligning and QC sequence per sample, performing short variant calls, doing joint call across populations, or filtering and QCing variants. And these are examples of GATK workflows that we have present in Docstore. There's plenty of other workflows you can choose from and search for. You don't have to get workspaces all set up on your own. We do help you get set up with GATK best practices on the featured page of Terra. So we have a list of featured workspaces you can kind of view and use if you like. And this is the featured or showcase and tutorials page. It offers curated versions of workflows and data packaged in a workspace. So there's the GATK showcase workspace, which is on the right hand of the slide. And this covers most of the GATK best practices. Then in the middle, we have the featured workspaces, which offers other created use cases that general Terra users wanted to share with other users, like DNA methylation or reproducible GWAS studies. In particular, there's a column to the left, which is the new and interesting part, which has new and interesting COVID-related workspaces that we'll look into a little bit later today. In particular, the GATK workspace examples on the right-hand column, they have downsampled data you can run as practice. We also have pre-configured workflows where the workflows are configured already, so you don't have to tinker too much with the workflows to run a data set. There's also time and cost estimates for running the workflows using the practice data sets or the sample data sets. So you get an idea of how much it costs to run the workflow with that given input size. So let's talk about the analysis mode. So we talked about bulk analysis. Now let's move on to interactive analysis. After running workflows on your data, you'll most likely want to interact with your data through plots, plotting it, or doing graph statistical analyses. And this is possible through the in-app Jupyter Notebook. Some details about the Jupyter Notebook that might be interesting is that you can run statistical and visualization analysis on your data in real time. You can perform analyses using a Python or R commands and can be easily shared amongst your colleagues. And Terra includes templates and showcase notebooks to help you even if you're not a coding expert. So similar to the featured workspaces with these workflows that are pre-configured. There are featured workspaces that have notebooks that allows you to kind of rerun GWAS notebooks that uses Hail or some other tool for notebook analysis. Now, with such a comprehensive platform, you might be wondering how you can ensure your collaborators and others can reproduce your analysis. This is where Docstore comes in. Docstore has a bunch of images. In the previous slide, you've heard about containers. Containers are a standardized way of computing, it's a way to standardize computing environments, making it easier to set up a computing environment and ensuring reproducibility. I'm sure many of you had to work with the same data on more than one computer, and you've run into problems with either the version of the software or having the dependency, needing to have the right dependency for the right version of Java. This hinders your progress, and if you don't have the right version, you might have different results. And containers eliminate these issues. A Docker container is a package that holds software and all its dependencies. Docker takes care of the software requirements. It makes it easy to set up the right environment with the dependencies you may need without having to worry about it. They're kind of like virtual machines with isolated compute environments where you can control the software installed. And by controlling exactly what version of GATK, Picard, and BWA you are using, it enables you and others to use the same version to reproduce your results. And containers are used both in the Jupyter Notebook and also in the workflows. So within the workflows, you have for each task or each command, you'll give it a Docker container you want to use for Whittle. And when you're running a Jupyter Notebook, you'll need to first create a virtual machine to run the notebook. And before you do that, you need to provide a Docker container for that notebook to run on. And in that notebook, you can list the specific versions of tools that you want to use. And once you have a Docker container, you can share this with others, and they can use the same Docker container when they execute your workflows or run a Jupyter Notebook. You're likely not doing research in a vacuum. You'll use workspaces normally to streamline collaboration and sharing. So workspaces are shareable, and you control with one click how much access each collaborator has. By default, a workspace is private to the person who created the workspace, but you can share it with others and allow them to either have read access, writer access, or ownership access, which allows them to share the workspace with others. There's also built-in security. Terra uses authorization domains for workspaces that include restricted access data. Only people within the authorization have access to that workspace. So as an example, let's say you've created a workspace and you share that workspace with someone else. That person would normally have access to that workspace. But now let's do that again with a workspace that has restricted access data. And with this kind of workspace, it normally has it's under a restricted access domain. So you would have access to that restricted authorization domain, and you would have access to that workspace. If you shared this workspace with someone else who doesn't have the authorization domain, they wouldn't be able to see that workspace unless they ask the person, or there's a site or a link it provides to the user as a person to contact in order to obtain access to that workspace. And the whole point of this is to prevent a user from unknowingly sharing a workspace with someone else who isn't supposed to have access to see the contents of that workspace. A bit more on security, a bit more on security. Here are some details about the security and privacy and how they're built into Terra. Terra is FISMA moderate, which allows Terra to host NCI and NIH controlled access data. It also leverages securities in GCP, Google Cloud Platform, allowing it to be FedRAMP authorized. And if you're not familiar with either of these, they're both of them use the National Institute of Standards and Technology guidelines to assess the IT framework of any organization working with government data. FedRAMP does contain controls above NIST baselines that addresses unique security elements needed for cloud computing, because Terra is a cloud platform. So we take security seriously when developing Terra, and these certifications are a show of it. Now now that you have an understanding of the benefits of working with Terra, let's look at the cost. Okay, so here's the big picture. Terra itself won't cost you anything. All the tools are free and open access, and there's a community of users and developers to help you and support you. There is a cost to the Google Cloud Platform. Terra as a UI doesn't cost anything, but you do have to pay for the cloud infrastructure that Terra Platform is built on top of, which is Google Cloud Platform. Thus, you pay for the cost of Google storage, Google Compute, and egress. The paying for it is easy. We make it easy by allowing you to set up a link between your Google Cloud Platform billing project and your Terra account. So Terra will know which billing account to send charges to after the initial setup. And later on, when you're running workflows or in Jupyter notebooks, Terra, your workspace, automatically knows which billing account to bill to. Right, so now that you know what you can do with Terra, let's talk about where to get started after you create an account. Here are some links to some useful sites to go to. I think this PowerPoint will be shared later on if you're interested in clicking on them, but there is a documentation page where you can find documentations on how to work with Terra, a lot of quick start documentation to get you started. There's also on the bottom here, 2 very useful featured workspaces that can help new users get accustomed to Terra. It allows you to work with workflows with varying degree of complexity. There's also a notebook featured workspace, a notebook quick start, which allows you to browse and access subsets of data in the Terra data library, import a cohort from the data library into your workspace, and perform some kind of interactive analysis with Jupyter Notebook. And on the right column, we do have a community-driven forum that you can post questions to. For the most part, we do have supports, like dedicated support team that answers these questions, so you're not like left out in the rain. There's also, if you're interested, a Terra landing page where you can get additional information about Terra. Hi everyone again. Welcome to the hands-on tutorial portion of the workshop. In this tutorial, you'll be working through the COVID-19 workspace. The workspace has several workflows, that you can that can be used to process COVID raw UBAM sequences. But for this tutorial, we'll focus on the first workflow, which imports a UBAM sequences into the workspace. And then we'll also work on importing a workflow from Docstore to validate the UBAM that we imported. Later, we'll also run a Jupyter Notebook on the COVID sample data as an example of performing QC using Jupyter Notebooks. The workspace has several other workflows. It has several other workflows in the workspace. So I would advise you to explore them further. That's very interesting, and we'll talk a little bit about it later on. The main objective for this tutorial is to show you how to clone a workspace, and how to run a workflow, how to import a workflow from Docstore, and finally, how do you run a Jupyter Notebook. Okay, so the first step in the hands-on tutorial is to clone the COVID-19 workspace. And to do this, you first click on the link provided in step 1, and that link will take you to the COVID-19 workspace. Once you're here, this is the workspace that some Terra collaborators created. The owners are listed on the right-hand side panel under owners, and there's some description in the dashboard description that they provided. What we want to do here in this workspace is create a version for ourselves that we can run workflows on, or yeah, we'll run the workflow in the Jupyter Notebook. To do this, we'll clone this workspace by heading to the top right, and you'll see this 3 dots in a circle. Click on that, and then you'll see the option to clone. Click clone. Once you click on clone, this pop-up window will appear with a few options you need to fill in, or a few boxes. The first one is the workspace name, and here it's asking you what you would like your version of the clone workspace to be named. We normally advise to enter in your name or initials, sorry, and anything other, any other identifiers. So I'll choose, I'll put in Barry and test. Next, we'll have the billing project, and this is where you indicate to Terra which billing project you'd want to charge to. Right now, I have several, but like you all, I have a billing project called BCC 2020 AnVIL Terra, and this is what we'll be selecting for this hands-on tutorial. In the next box, we have a description. And in the description is the original notes from the author, which is really the dashboard contents. We don't need to edit anything here. We'll just leave it as is. And here you see there's the authorization domain I mentioned earlier. We won't need any authorization domain, or you don't have to worry about that for this workspace. You'll simply need to clone this workspace after you filled out the name of the workspace and the billing project. So go ahead and click on Clone Workspace. And after you clicked on Clone Workspace, you'll be automatically taken to your version of the clone workspace. And you can identify this by going to the right-hand column or panel, and under Owners, you should see your account name. And also your naming convention will be listed in the workspace name up top. And before I go any further to step 2, you don't need to put anything under the authorization to name. You can keep, you should keep that empty. Okay, so I'll go ahead and move on. And next, we'll talk about the workspace. So welcome to your first Terra workspace. This belongs to you now. And like we mentioned earlier, the landing page of a workspace is the dashboard, where you have the description of what your workspace does. And here it copied over the original contents of the original COVID workspace. So this is what the authors previously listed down. And for future reference, if you want to edit anything in the dashboard, you click on this pencil button here. Next, we have the data tab, and this again lists the data tables, and the tables are listed here with sample and sample sets. What's very important here is the sample table, where each row is an individual COVID sample, and each column is a descriptor of that sample. So there's the biosession number, the SRA ID, library ID, the run date, and other useful metadata. Next, we have the notebook, and within the notebook, we have the COVID-19 QC notebook, which we'll be running. There's also a list of workflows in this workspace. And lastly, the job history, where it lists all the workflows that you've executed in your workspace. Currently, there's nothing here because this is a brand new workspace that you clone for yourself and you haven't run anything yet. Okay. So an overview of the COVID workspace. The COVID workspace was created to help researchers who don't have access to strong lab resources obtain access to publicly available raw COVID sequence data. It also provides some suggested workflows to process raw sequences. The workspace has a combination of workflows and Jupyter notebooks to allow users to download COVID sequences from other repositories using the SRA session ID from the NCBI archive and perform read alignment on those sequences. And once those sequences have been aligned, you can perform some QC on those alignments, those BAM files using a Jupyter notebook. And lastly, after identifying which samples are valid through your Jupyter notebook, you can then later run a workflow that generates a JSON file, and that JSON file can be given to a tool called Nextstrain, which is a web browser that allows you to create phylogenetic trees. So in step 3, we will create a virtual machine for our Jupyter notebook. And to do this, you go to your workspace and go to the top right. The top right, you'll see play button, and click on that play button, and this will cause a panel to appear on the right-hand column. After clicking this, you will then be able to select the parameters for your virtual machine that will run the Jupyter notebook. There's the chance to write in or enter in the Docker container. For this notebook, the default Docker container is all you need, along with the default profile for the compute usage. Now, if you wanted to change something up, perhaps you wanted more virtual machines, or sorry, not more virtual machines, but more CPUs or memory, you can use the custom option here and individually select different types of CPUs or memory. But again, for this tutorial, the moderate or the default is all you need. Just going to look at the chat really quickly.
They were asking if Jupyter notebooks always require a virtual machine.
Yes, you will need the virtual machine in order to run a Jupyter notebook.
I can expand. The virtual machine is asking for resources from Google Cloud Platform to analyze your results.
Correct. And if you are only viewing the contents of a Jupyter machine, you don't necessarily need to be, you don't need a virtual machine. You can not create one and just view the contents of the Jupyter notebook. Okay, so the default settings are all right here. And after you're ready, just create the virtual machine here. Okay, and once you select that, you'll see something that says a notebook runtime is being created. So we can leave that sitting for now. We'll use this notebook later on in the tutorial. That's refreshing. I'll move on. Okay, so step 4. There are several workflows in this workspace, but we'll focus on the first workflow of this workspace. And just to go over it really quickly, there's a nice, pretty diagram in the dashboard with the listed workflows. So there are a few workflows listed here, but the main kind of process in this workspace are the column on the right. Each green box here represents a workflow. So for the first workflow, it's called fetch SRA to BAM, and simply you provide it a SRA ID, and it will download a UBAM from the NHBI archive. Then with this UBAM, normally you would do an assembly using this workflow. This workflow would provide some input with the BAM file along with some FASTA files. And you can perform QC on these files using the Jupyter notebook. Once you've decided which ones are high quality, then you can move on to this other workflow called build tree, which will create a JSON file that can be plugged into NextTrain, and NextTrain creates this phylogenetic tree. And just to see what an example of this phylogenetic tree looks like, at the bottom here, so you'll get a nice image. But for this workflow, or for this hands-on tutorial, we're focusing on the first part, and that's SRA, giving a workflow an SRA ID and importing a UBAM. So to run this workflow, we will start with, head to the Workflows tab. And once you're in your Workflows tab, we will select the Fetch SRA to BAM workflow. And before we move on, I just wanted to show that all of these workflows are being imported from Docstore, as mentioned by the source in the workflow card. But anyway, click on the Fetch SRA to BAM card. And once you click on that card for that workflow, you'll be taken to the workflow configuration. And once you're in the workflow configuration, we'll change a few things to run it for this hands-on tutorial. The first thing I'd like you to do is, for version, you'll click on DP NextTrain. This is where there's different versions of the workflow, but for this tutorial, we're interested in DP NextTrain. Once you selected that, we'll move on to the next part. And here you have two options. You can either run the workflow with inputs defined by file paths, or run with workflows with inputs defined by data table. With the first option, it's saying that you're not going to use the data tables that we've listed in the workspace. Instead, you're going to provide the inputs manually for this workflow, and the inputs are here. But since we have the data tables already, we'll select the second option to run the workflows using the data table. And once we've selected the second option, there's two more options that pop up. And the first one is to select the root entity type. And by root entity type, we're just saying which sample or data table you want to use. There's currently two options for this workspace, and that's sample and sample sets. And just to give you an idea of what this is referencing, you don't need to do this, but I'm going to go to the data tab and a new browser tab just to show you that the two available tables that are inside our data tab is sample and sample set. So you're telling the workflow which table to work with. And currently we'd want to work with the sample table because that's where our accession for each sample is, SRA ID. So for the select root entity type, select sample. So now that it knows which table to work with, we'll then indicate which row within that table to select. So you do that by selecting data, and then this pop-up will appear. You can choose to process all 68 rows within that table, or choose specific rows. And we, in this tutorial, will just choose three, the top three available here. So to select a row, you click on the checkbox on the very left-hand side of each row. I'm going to select the first three. Once selected, you go to the bottom and click on OK. Okay, so now we've selected our three rows. Next, we will enter in the input variables for this workflow. And to do this, it's at the bottom here. And I just wanted to show you really quickly that there's three tabs here. There's scripts, inputs, and outputs. Scripts is the actual view of the workflow that you're running. You won't be able to edit it, but you will be able to view the workflow, the WIDO workflow script here. And the input is the inputs you need to run this workflow. And here we have three inputs. The first one, as mentioned on the right, is required, while the other two are optional. And the first variable here is the SRI ID. And if you, now that we've so far, just a reminder, we told the workflow which table to use, which rows to use, or which samples. And now we need to indicate to the workflow where does our SRA ID exist within that sample table. And the SRA ID exists under the column name accession. So for this attribute, for this variable, we'll start typing in accession, and here we'll get the column name listed in the drop down. Now it's important to have the beginning part, this dot, which indicates to the workflow, again, that anything after this dot is a column name within the data table. Okay. So in this section, again, this dot accession. Next, we'll move on to the Outputs tab. And in the Outputs tab, we'll do something really simple or easy because there's a lot of outputs being generated by this workflow. We'll just click on this, clickable font here called Use Defaults. Once you click on it, the values or attributes for each output variable are listed or automatically updated. And what we're essentially doing here is we're indicating or telling the workflow that once the workflow has completed, for the outputs that are generated by this workflow, we want you to save the outputs or place those outputs within our data table under the column name file sample session or library ID or whatever other kind of column names are listed here. And now that we've added these in, lastly, we'll want to save this. We've done our configurations for our workflow. The last step is to run the workflow. And to do this, you click on Run Analysis. Once you click on Run Analysis, a pop-up will appear asking, or it's like confirmation window, whether or not you want to run this workflow on the 3 samples that you selected, and we'll click Launch. Yeah, we do. Once you click on Launch, you will automatically be taken to the Job History tab. And again, this lists all the workflows that have been executed, including the ones you just executed now. So for me, I've selected 3 of the rows or 3 of the samples, and each one is a workflow execution in itself. So I have 3 execution statuses. On the status column is where Terra will update you on the status of the workflow. Right now it's queued, meaning it's kind of being sent to the job manager. There's a few other ones. I'll refer to the tutorial. There's submitted, where the JSON inputs are being sent to the virtual machine. Then there's running, where the job is actually running in the Google Cloud machines. And lastly, there's done and done but failed, which means something went wrong and you'll need to troubleshoot. And if you would like to troubleshoot if something went wrong, you can select this view icon to view what went wrong at what task within the workflow failed. Okay, and you can see the status is automatically updated. So at this point, mine is at the submitted status. So this might take about 3 to 5 minutes to complete. In the meantime, we can go over any questions that you may have. Mine have succeeded. No, you won't be able to get a cost estimation before running a workflow. The cost is, that total run cost is filled in after running the workflow. And I think it might be also restricted to certain billing projects because of a setup being done with BigQuery. So, yeah.
So yeah, currently only road-based billing projects can report the cost within the Terra user interface. However, one of the best practices for these types of featured workspaces is that the dashboard, which gives you the overview, may include some cost estimates from the original authors.
Thanks, Beth. And also thank you for responding about running. Yeah, I think I ran these workflows before, and there is this call caching option, which automatically reruns or it doesn't run a workflow if it's run already. Instead, it copies over previously run workflows and imports the output generated. And that's useful just to save on money. And there's also, if you don't want that enabled, if I go back to the workflow, you can turn off use call caching.
Barry, I have a question. If you're using call caching in that manner, are there cloud costs incurred? Is anything being computed?
I don't believe so, and it's also in different stages. So call caching will run if a workflow failed in the middle. It will call cache for the task within the workflow that completed. So no charges would be incurred. But afterwards, if there are parts of the workflow that failed, and at that point, cost will incur. All right, so I'm going to move on. For the most part, I'm going to be going over currently how to look for the outputs generated. And we won't necessarily need to see or have all of these completed at this point. So let's go ahead and start that. Okay. Okay, so after a workflow has been run, you'd want to find your results. And to do this, you'll go back to the data table. And I'm going to show you something real quick. You don't have to follow along at this point. I'm going to go back to the workflows table. And just to remind you, for the workflow configurations, we use the defaults setting. So the outputs generated by this workflow would be available in the data table under these column names. So what I'm going to do is go to my data table and look for these column names for the results or the outputs that were generated by the workflow. So if everyone can all go to the data tab. Once that you're in the data tab, click on sample. And once you're in the sample table, you have, again, a list of rows or COVID samples listed. And what we want to do is click on this gear icon. And in the gear icon, I'm going to unselect all of these column names by clicking on none and selecting only the ones that I'm interested in looking at. And those are the accession IDs and the read UBAMs. And these read UBAMs are, again, the outputs that should be generated by the workflow. I'm going to click on done. Once I do that, we have three files, 3 UBAMs generated for the first 3 rows. And that's because I selected those 3 rows. And here is my outputs. There's also some, if you remember, there's also other outputs generated, and that's in other columns if you want to select them. Furthermore, so once the UBAMs are here, next thing you'd want to do is perhaps download them. You can click on this UBAM link, and you have the option to download this file locally, and it also indicates how much it would cost to download the file listed here. And also there's the actual location or the file path listed in this terminal download command. So the file path where this file actually exists is inside your workspace storage bucket, And this is the actual path for it if you're interested for further analysis later on. But I'm not going to download anything. I'm just going to click on done. And as a reminder, these tables are metadata information. So it's not like files exist here, but these are simply the links to those files or the file paths. So if I were to click on this pencil edit button, it will only list the file path to that file. Okay. Okay. And lastly, I wanted to go over a different way of looking at the outputs. So if I go back to my job history, or actually let's go to the workflows really quickly and go to our fetch SRA to BAM workflow configuration. Now remember, we've selected the second option, which says to use the data table to take in input, and it also indicates where we want to put our outputs. But let's say, in case you chose the first option, where you manually entered in your data, then in that case, you wouldn't be able to link to the data table because you've manually entered in your input data. So in that case, this output table would be useless when you run it with the first option. So instead, what you could do is go to the job history, go to fetch, then click on view for one of your executed or succeeded workflows. And this will open up the job manager. And here you can have, or you can see the inputs that were provided for this executed run, and also the outputs that were generated, all the outputs. And this is our UBAM link too. So you can click this to download it also as well, but I won't do that. But again, this is just another example of getting the outputs from an executed run. Okay, so I'm going to close out this job manager. We're going to move on to the next step in the tutorial. So with step 5, we are going to play a little scenario where you've downloaded the UBAMs and you want to perform an additional analysis that isn't provided in the listed or the given workflows here. Or you're creating a new workspace and you want to import those workflows into your workspace. So what you want to do is click on find a workflow. And once you do that, this window will appear. It will first give you a few suggested workflows that you can work with, but what we're interested in doing is importing workflows from Docstore. So to do this, I'm going to right-click on the Docstore link and open this in a new tab. Once I do this, I'll move over to the Docstore site link that is open. And on the left-hand panel, I'll search for validate-bam, which is a workflow that we'll want to import in. And one thing to note here is, since you're you're being directed from Terra, which only uses WDL workflows, there are pre-filters or filters that are already enabled to only search for WDL. So just an easy way of searching things. So after you type in validate-bam or validate-bam, the results will show this workflow called validate-bam from JTK Workflows repo. We'll select this workflow by clicking it. And once you click on it, you'll be taken to the validate-bam workflow public view with all the details about this workflow. And what we'll do is click on AnVIL, or go to the right-hand panel with the box that says launch with, and there's different platforms you can launch this workflow with. We'll choose AnVIL. And again, AnVIL is a flavor of Terra, or it's being powered by Terra. So that's why you'll It's essentially Terra, but in AnVIL version. Once selecting AnVIL, a new browser window will appear. And here it's asking you where you want to send your workflow to, or what workspace. In the first box, you'll enter in the name that you want to name this workflow within the workspace. And I'll keep it as is. Next, I will add the destination or the workspace I want to send this workflow to. For this, I will start typing in the name of my workflow. So that was COVID. Oh, wow. -19. Copy. Berry. Okay, so I have COVID-19 copy berry test. This is the clone version of the workspace that I made. I'm going to select this. Next, I'm going to import it into that workspace. I'll click on import. And once I do this, I'm taken to my clone version of the COVID workspace inside the workflow configuration page within the workflows tab. And now we'll want to set those workflow configurations for this validate BAM workflow. For this, the version, we'll leave it at master. And for the whether or not we want to use the data table or not, we'll select yes, and use the sample table. Again, because that's where our UBAMs are, and that's what we're trying to validate. Next, I will click on select to choose those specific rows that has the UBAMs in them. And that's the first 3 rows for me. And next, I will click on OK. So after clicking on OK, next we'll fill out the input variables listed here. And again, there's a lot of variables here, but the only thing that's required is the BAM file, which is the first row. And again, here we want to tell Terra under which column are the UBAMs that we want to validate. And those UBAMs are under this read, this dot read UBAMs, right? Lastly, I'll go over to outputs and select use defaults and click save. Okay. So we're ready to launch this workflow now. You'll want to click on Run Analysis. And this window will appear, and click on Launch. And again, we have 3 samples running, and it's on queued. But this workflow might take a bit longer than the previous one, so I'll leave it here for now. But yeah, after running the workflow, it will complete, and we can move on to the next workflow. But in this case, what we wanted you to understand is how to import Docstore workflow into your workspace, which you've done, and you've run that workflow, which is a bonus step. Okay, so we should be on the next step now, which is running the COVID-19 QC notebook. So we're at the step of running the COVID-19 data QC notebook. And to do this, we'll go over to the notebooks tab. Inside the notebooks tab, we have one COVID-19 Jupyter notebook. And to run this, you click on the notebook itself. And once you do this, you are automatically sent to the Jupyter in preview mode. There are different modes of viewing this notebook. There's preview, where you can view the contents of the Jupyter notebooks, and you don't need a runtime or a virtual machine for it. And there's the playground mode, where you can edit or you can execute the codes within the notebook, but any edits that you make within this notebook session won't be saved for the future. And then lastly, there's edit mode, where you can actually edit the changes and the results that you've created. And for this tutorial, we'll just go ahead and use the playground mode. And again, there's a little bit of warning that says, hey, if you use playground mode, none of the changes that you've made to the notebook will be saved, and that's okay. So click on continue. Now, so one other thing to note is, again, the notebook is being run on the virtual machine, and earlier on, you created that virtual machine, so that's what we'll be running on. And during the time of running the workflows that we did earlier, which took a bit of quite some time, Terra will automatically pause your virtual machine so that you're not incurring cost while you're not using it. So right now, the virtual machine that we created for this notebook is being created again. You can tell by the starting notebook runtime environment. We're just waiting for that right now.
So we could use this time to talk about some questions from the chat. Even though I did try to answer them there. Is there a difference between Terra and AnVIL powered by Terra? And the answer to that one is no. They have the same backend, so they have the same features. It's just kind of different skins. And also, if it would cost, if there would be cost differences if you run the same workflow with different samples. And in this case, yes. For this workflow that we're using, if you pull a UBAM file that has, for example, higher sequencing depth, it's a larger file and would cost more to copy.
So once your virtual machine is running, it will indicate that on the top right, and you'll be taken into the Jupyter notebook, and you're ready to run cells. If you're not familiar with a Jupyter notebook, this is what it looks like. Jupyter notebooks are kind of divided into cells. So there's different types of cells. There's a markdown cell where you'd write descriptions of your analysis. So these two are examples of markdown cells. And then there's these code blocks. This is where you actually enter in the code that you want to run. And in this case, we are running on a Python kernel, as indicated at the top right here. So we have some Python code where we're importing valuable kind of packages that we need for analysis in the future cells of this notebook. Okay, so and to run a code cell, you will click on that code cell, and at the top here, there's a run button, and you can run that notebook or run that cell. And in that case, a star will appear between these brackets, indicating it's currently running. And there's also a digit that appears after it's run, indicating that it's completed. Next, so in this step here, we're importing in packages. In the next step, we're going to set up our notebook environment and adding them into variables so that we can quickly access them later on in the notebook. And in this case, we are getting the billing project, excuse me, for this project and the workspace name, along with the associated the Google bucket associated with this workspace. They're all listed here. So I'm going to go ahead and run this cell again, or as well. And moving on to this step, which is load output data. So in this cell, we are downloading the data table that's in our workspace, in our data table in our workspace, so that we can later use the metadata within each of these columns to perform some QC steps. So by entering, running this code, we're importing in that data table. So I'm going to run that code. now we have our data table imported into our notebook. And as a reminder, the data table I'm referencing is, you don't need to follow me for this, I'm just showing you. If I go to the sample tab or data table in the sample table, this is what we're bringing into our notebook. So here, this is my sample table, and as noted by the 3 read UBAMs that I have listed here. Okay, so now that we have our sample table in our notebook, we'll start doing, performing plotting scripts. So the first thing that we'll do under the plotting step is import library for plotting, in this case, matplot library. So I'm going to run that cell. And next, in the next cell, we're going to import the certain files or certain data from our sample tables and put it into a Python data frame. In this case, we're creating a data frame called assembly length that will hold all the sample information under the column assembly length. And we'll do the same thing for mean coverage for each of the samples. So I'm going to go ahead and run this. And here we have our table listed in this output cell. And in this particular output, we are displaying the mean coverage, so the second data frame. And in this data frame table, we have for each row, each of the individual samples, and then specifically the mean coverage for each of those samples. And again, this is metadata that was brought in or that already existed in the workspace. Okay. Any questions so far?
One question from the chat is, where are we importing the matplot library from? And I believe it is pre-installed in the default Docker container that you selected when you did your notebook runtime configuration.
Yes.
So you did not have to first install. You could just go directly to import.
Yep. Okay. So next step is to plot our data frames. And here's the funky part. So for the original analysis that the workspace authors did, I think they're working with a few samples, maybe 10 listed here. Since we brought in all of our 68 samples in our data frame, ours is going to be a bit cluttered, but we're going to go ahead and plot the data within our data tables by running this row or this cell. Okay. So here we have 2 plots that are generated, one for assembly length ambiguity or unambiguous, and the other one being mean coverage. The first plot being assembly length ambiguous reads. So for each sample, we are measuring the number of reads that are unambiguous. So that means these reads are solid. They map to the reference very well, and there's no ambiguity as far as mapping to another part in the reference. So that's very good quality. For any samples that have low unambiguous reads, chances are that's not something you want to use for in our next step when we create our phylogenetic tree. And then another kind of basic QC for next generation sequencing is mean coverage, which is the amount of coverage for read sequencing. And the lower coverage is generally not very low quality. It's very low quality. But in this case, we have one sample here that's skewing the whole plot, which has a tremendous, very large amount of mean coverage. So it's causing everything else to look, horrible. But so a next step in this analysis would be to remove that and see what the other samples look like without that large sample skewing everything up. And yeah, after we've determined which samples that are low quality and which one are the best quality, we can use those samples to, in our next analysis, which is creating that phylogenetic tree, to choose specific ones and provide those good, valuable samples to next train to create that phylogenetic image. And that is it for this particular notebook. So with the end of that notebook hands-on tutorial, that wraps up this tutorial in general. And I think, again, recap what we were trying to teach you here. And that's how to clone a workspace in Terra, and how to run a workflow, how to import workflow from Docstore, and showing that integration between Terra and Docstore. Finally, how to run a Jupyter notebook.